Hi, Professor Stefero here, and today's screencast is on the fluid mosaic model of the plasma membrane. This model was described in 1972 and explains the structure of the plasma membrane. The figure on the left is a TEM of the plasma membrane, and the figure on the right is a representation of this membrane. Recall that the main components of the plasma membrane are phospholipid molecules. The plasma membrane has two layers or a bilayer of phospholipids that form the plasma membrane, and we symbolize these with the spheres with the tails attached. Taking a look at these molecules, the heads are hydrophilic, polar, and water-loving. The polarity is due to the charged phosphate group associated with the head. These are the portions of the molecule that are found on each side of the plasma membrane facing the watery environment of either the cell or the cell's exterior. The core of the plasma membrane consists of fatty acid tails. They are hydrophobic, nonpolar, and water repelling. This part of the molecule provides the fluid portion of the fluid mosaic model. So let's take a look at the short animations about plasma membrane structure.
The plasma membrane is composed of a bilayer formed by two chains of phospholipid molecules. The fatty acid tail of the phospholipid molecule is nonpolar, whereas the head of the phospholipid molecule containing the phosphate group is polar. The two phospholipid molecule chains layer like a sandwich, with the fatty acid tails forming the interior of the membrane and the phospholipid heads facing the watery environments found inside and outside the cell. Cholesterol is also found in the plasma membrane. It helps to stabilize the phospholipids by preventing the fatty acid tails from sticking together. Proteins that span the entire membrane form the selectively permeable membrane that regulates which molecules enter and which molecules leave a cell. Carbohydrates sticking out from the cell surface identify chemical signals and prevent the cell from succumbing to infection.
So what does it mean for the membrane to be selectively permeable? This describes how substances cross the plasma membrane to either enter or leave the cell. And generally we can describe two methods. One method is through the lipid bilayer itself. Molecules that pass through the lipid bilayer tend to be very small molecules or nonpolar or hydrophobic, like other lipids or hydrocarbons. Let's take a look at the animation that describes this.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So how do charged, large hydrophilic molecules pass through the plasma membrane? It's by the second method of transport, through protein channels or protein transporters that are found floating in the bilayer like icebergs. Here in the animation you see the purple blobs representing those proteins. These create hydrophilic passageways, allowing ions and polar molecules to enter and leave the cell. Other proteins in the membrane will serve as pumps, using energy to actively transport materials into or out of the cell. Let's take a look at the various kinds of protein carriers that are found in the plasma membrane. The first of these integral proteins works as an ion channel. The protein is a charged pore that allows molecules to pass through the cell membrane if they are the correct size and the correct charge. Second are membrane pumps. Membrane pumps are carrier proteins that use energy to move molecules and ions into and out of the cell. The sodium-potassium pump is an example, moving sodium and potassium using ATP energy to allow for that movement. The next are carrier proteins. Each is built to bind and lead a specific substance into the cell. Carrier proteins bind to this particular solute and lead it into the cell. The protein will change shape as a way to move the molecule. Last are the receptor proteins. These bind messengers or hormones that change the permeability of the cell. They'll trigger the formation of another message in the cell, or they'll activate enzymes in the cell to speed up reactions. So those are the integral proteins. Okay, let's wrap up the structure of the plasma membrane. Remember, we use the fluid mosaic model as the basis for describing this structure, and you should be able to answer the following questions after you've watched the screencast. Good luck, and I'll see you in class.